Okay, I want to very warmly welcome everyone to the launch event of the Catalyst seminar series in children and young people's mental health. These seminars are the result of very hard work by the Children and Young People's Strategy Implementation Group, and they are generously sponsored by the UCL Neurosciences domain and supported by the domain together with the Institute of Mental Health. The purpose of the seminar series is to foster cross-disciplinary dialogue, innovation and collaboration across UCL, and also to showcase our strong research in areas that are relevant for children and young people's mental health. We've got a really exciting first event with speakers from very different research traditions, including population health, cognitive neuroscience, and basic neuroscience. And the speakers will consider the impact of early adversity on development. Each speaker has been asked to touch on how their research would benefit from inputs and collaboration with other disciplines. So we really want to get people thinking about these sort of connections that they can make across discipline. We'll start the event with a few words from Professor Alan Thompson, who's the Dean of Brain Sciences and who commissioned the UCL mental health strategy that has motivated these seminars. And we will also hear from Professor Tony David, who's the Director of UCL Institute of Mental Health, which is there to bring together people from across UCL to do strong mental health research. We'll then have 3 short talks, one after another. First, Dr. Rebecca Lacey, then Professor Eamon Mccrory, and then Dr. Andrew Macaskill. You're welcome to ask questions in the chat, and we will select a few of these for a discussion by the speakers at the end of the talks, but we won't be taking questions in between the talks. And we particularly like to encourage questions that are aimed at fostering cross-disciplinary dialogue. If you have very specific questions about the talks themselves, we might encourage you to get in touch with the speakers via email for those questions. It would be really good if people would ensure that their microphones are muted. And without further ado, I would really like to welcome Professor Alan Thompson to say a few words, followed by Professor Tony David.
Thanks, Essi. Well, as many people know, the UCL mental health strategy was launched in 2019, and the vision was to ensure that UCL is recognized as a world leader in interdisciplinary mental health research. So the key implementation goals of the strategy included a development of clear mental health community and presence, active support for interdisciplinary research that advances our understanding of different mechanisms that drive the development of mental health problems. And the establishment of the Institute of Mental Health, led by Tony David, is a key enabler of this strategy to raise profile, unify our diverse and excellent activity, and increase our productivity. So one key focus, led by Essi and her colleagues in the Strategy Implementation Group, is on child and young people's mental health. And activities within this area include early career initiatives and a new website showcasing research in this area at UCL. One reason why we focus strongly on this area is the increased public awareness of the importance of preventing and treating mental health problems during childhood and adolescence, as well as the recent substantial funding initiatives to encourage research in this area. And UCL clearly wants to be strongly represented on this front. The Catalyst seminar series, which has been launched today, is designed to build a stronger community and cross-discipline communication in children and young people's mental health within UCL. The seminar series, as Essi has mentioned, has been supported financially and administratively by the Neurosciences domain and the Institute of Mental Health. It goes without saying that we have brilliant scientists conducting research that is relevant to this area in a number of different fields, including basic neuroscience, cognitive neuroscience, computational psychiatry, clinical psychology, social psychiatry, epidemiology, and social sciences. However, all too often people have worked in silos and do not necessarily know about exciting and relevant research conducted in an area that is different from theirs, despite being in the same institution. So it seems to me that this launch event is a perfect example of how we can showcase research from different traditions and how we may benefit from connections across disciplines as we go forward. Although it's not ideal that we're not meeting and networking in person, we can't put everything on hold while we sort out this pandemic. So I hope that we get a really good head start with these monthly virtual seminars, which we can then build on when we meet again in person. Knowing Essi and our colleagues, I am sure this is going to be an exciting and informative series of presentations, and I wish you every success both for this series and the wider developments in child and young people's mental health. And with that, I'll hand over to Tony David, Director of the Institute of Mental Health.
Thank you very much, Alan, and thank you very much, Essi, for inviting me to say a few words. I'm absolutely delighted to introduce the Catalyst seminar series. It's really a very welcome initiative. There's two particularly good reasons to support this now, why the timing is particularly apposite. First of all, of course, this terrible pandemic that we're in, a virus that we're told spares children and young people, and yet they seem to be hardest hit by all the other methods and measures we've had to take. Children and young people seem to have been in the firing line when it comes to their education, their peer relations, travel, exercise, and physical health. So it just reminds us how important our young people are and how sensitive they are to both positive and negative influences. So the timing is particularly ripe because of that reminder that the pandemic has given us. The other reason that the timing seems perfect to me is slightly personal, and that is that the Institute of Mental Health is now about 2 years old since it was launched. And as Alan Thompson has said, it's a virtual institute that aims to bring together all of the people interested in mental health research across the university from different faculties and different disciplines. So after 2 years, I think the Institute of Mental Health has sort of firmly bedded in. Hopefully people know about it. And it's at a point of confidence that it can start going beyond its overall remit to showcasing what I would think of as the jewels in the crown of mental health research at UCL. There are many, cognitive neuroscience, basic neuroscience as applied to mental health, epidemiology, some of the many, but I think children and young people's research is particularly excellent here. And I think the seminar series shows why that probably is, because it does draw on such diverse disciplines that we're going to hear about today, from public health, epidemiology, basic neuroscience, cognitive science, and clinical psychology. So I hope that we'll have other groupings focusing on particular aspects of mental health, whether they're techniques or disorders, and that our umbrella of the Institute of Mental Health is wide enough to shelter and nurture all of these different disciplines. I think it's a particular challenge for children and young people because it does impact on so many areas. And indeed, when we look at UCL, there's clinical psychology, there's the Institute of Education, there's cognitive and basic neuroscience, as I've said, there's psychiatry itself, the clinical division, and child psychiatry, which finds itself in a completely different faculty from brain sciences for peculiar historical reasons. So making sure that none of these elements feels as though it's cut off from the rest is a very important endeavor. And the seminar series will, I'm quite sure, make sure that we get to be the whole to be more than the sum of its parts. So as an exemplar for, I think, how mental health research is going to develop, I'm particularly looking forward to this seminar series in children and young persons' mental health.
Thank you, Tony, and thank you, Alan, for this really sort of encouraging words. And I think without taking longer, we should kick off with our first talk, which is Dr. Rebecca Lacey, who is talking about consequences of early adversity from a population health epidemiology point of view. Each of the speakers will take just under 15 minutes, we hope. And as I said before, we'll do the questions afterwards in a form of panel discussion.
Okay, thanks, Essi. I'll just share my screen. Oh, it doesn't seem to work properly. Okay. Great. Can you see that okay?
Yes, it's great.
Okay, great, perfect, thank you. Okay, thanks very much. Thank you for this invite to speak today. This is fantastic. So my name is Rebecca Lacey. I'm a social epidemiologist who does life course research. So I use longitudinal population data sets, such as data from birth cohorts and studies like the Millennium Cohort Study that I'll be showing today. Very broadly, my interests are on how stresses in early life affect mental health right across the life course. And today I'm going to be presenting one piece of work, which is part of a bigger ESRC funded project, which is looking at the clustering of early life adversities and life course mental health and inflammation using longitudinal data. The particular piece of work I'm going to show today is focusing just on the early part of the life course. And it was a piece of work that was led by Leo Bevilacqua, who was the research fellow on the project until fairly recently. So big thanks to him for this work. So early life adversities, which are sometimes called adverse childhood experiences, or ACEs for short, are associated with a range of poorer health outcomes, at least at a population level. And by the term early life adversities, we're typically referring to stressful experiences in childhood and include experiences such as child maltreatment and also indicators of household dysfunction, such as family breakdown or parental substance misuse or mental health problems. A lot of this research has been conducted in adult populations, so often asking adults to recall information from many decades before about things that happened to them, which clearly has some problems. Health problems as a consequence of adversities are likely to emerge closer to the time of the event, and by using longitudinal data from cohort studies from early life, we're able to look at when differences in mental health might emerge. And there have been some previous studies which have looked at externalising, internalising behaviours in young people and young adults, but we know less about how adversities impact on the development of more adaptive behaviours, such as prosocial behaviours. So prosocial behaviours are important for later educational outcomes, well-being, and also for things like adjustment. Also, one of the main focuses of this whole project is on how early life adversities are considered in analyses. And there have been many studies which construct sort of a cumulative risk, or an ACE score, which is just simply the number of adversities that have been experienced. And the number of adversities are typically related in a graded fashion with whatever health outcome that you're looking at. This only tells us so much about the mechanisms, for example, that are involved, and also doesn't give us really any information on what adversities that person has experienced in combination. One alternative is to look at what adversities somebody has experienced, to just look at specific individual adversities and to see if specific adversities are driving associations with health. There are clearly lots of other ways of dealing with adversities and analyses, including using methods that account for clustering. And this is actually one of the main thrusts of my research project, but it's a whole talk in its own right, as I'm sure you'd appreciate. So I won't be saying more about this unless people are particularly interested in hearing more about that aspect at the end of the talk. But just in this project, we're looking at this particular paper, we're looking at ACE scores and also individual adversities as well. So this particular paper has 3 research questions. So firstly, are early life adversities associated with development or change in internalising, externalising, and prosocial behaviours across childhood and into adolescence? Secondly, whether particular adversities were particularly harmful. And then thirdly, whether there were any gender differences in these associations. So we've used data from the Millennium Cohort Study, or the MCS, for this paper. The MCS includes information on around 19,000 children and their families born in the UK between 2000 and 2002. It oversamples often underrepresented children, so those living in more deprived areas, children from ethnic minority backgrounds, and those in the smaller nations of the UK. It's a multi-purpose cohort study, so it includes very rich information on physical, socio-emotional, cognitive, behavioural development, along with a whole host of other things. There are currently 7 sweeps of information available. The first was at 9 months, and fairly recently the age 17 data has been released. This particular study is using information on adversities in very early childhood periods, so up to age 3, in relation to trajectories of mental health from age 3 through to age 14. This analysis was done prior to the release of the age 17 data. So these are the adversities we've looked at. So again, as I mentioned, we've looked at this very early childhood or infancy period, and they're largely reported by the child's parent, which most often is the mother in the Millennium Cohort study. So we've looked at parental separation or divorce, parental depression and anxiety, parental drug misuse, parental alcohol misuse, interparental violence, parental discord, harsh parenting, and physical punishment. I can give more information at the end on what we consider to be the sort of thresholds for adversity for each of these if anybody has queries about that. In terms of our outcome, we've used the Strengths and Difficulties Questionnaire, or the SDQ, which was available from age 3, 5, 7, 11, and 14 years. This is a very widely used scale. We've constructed externalising, internalising, and prosocial scores in the usual way, from the peer problems, emotional problems, indicating internalising problems, conduct and hyperactivity relating to externalising, and then the prosocial score by itself. And then we've also included some covariates, for example, gender, as that was one of our research questions, but also some socio-demographic information, like things like indicators of socioeconomic position and ethnicity. So we've looked at 2 different ways of operationalizing ACE measures in the study. So we've constructed the ACE scores, which is the total adversities reported by age 3. We've also looked at this accompanied by the individual adversities, which allows us to see whether specific adversities are more harmful than others. In terms of the statistical analysis, we've used multi-level linear growth models to test associations between adversities and then the 3 trajectories of internalizing, externalizing, and prosocial behaviors. These types of longitudinal methods allows us to count for dependency of observations within individuals, so the repeated measures that we have, and it allows us to look at change in the 3 outcomes from age 3 right through to age 14. And our sample for each of our outcomes is around the 8,800 mark. So firstly, looking at the prevalence of early life adversities in the MCS, that's just showing the percentages of children who experienced each of the adversities. So on the left-hand side, the green bars are for the boys and the turquoise are the girls. The most commonly reported was parental depression or anxiety, which is probably no surprise, sadly. So that's around 37% of girls and boys, followed by harsh parenting, which was 30% of boys and 25% of girls. Parental separation and parental drug use were the least commonly reported. On the right-hand side, we have the ACE scores. So only 25% of boys and 27% of girls had not experienced any of these things by age 3. And then the more extreme end, we have 19% of boys and 17% of girls had an ACE score of 3 or more. You can see there are some very small gender differences shown here. So boys are more likely to receive harsh parenting and physical punishment than girls, but we actually found no gender differences in the rest of the analyses. So from this point on, I'm going to be showing boys and girls together. So moving on to look at the associations between the ACE score, so the number of adversities, and the 3 trajectories that we've looked at. At the very top here, we have the internalizing behaviors. So you can see that internalizing behaviors increase from age 3 through to age 14 for all children. The red line at the bottom is those children who have 0 adversities. The pale grey line is those who experience 1, the yellow is those who experience 2, and then the purple dashed line is those experiencing 3 or more. And you can see already that by age 3 that there are these graded differences in terms of internalizing behaviors, but we also get this divergence over time as well, so that those children who experienced any adversities had a steeper increase in internalizing problems relative to the children who had experienced none at all. In the middle, we have externalizing problems, which you can see decrease with age for everybody, but you get this sort of slight kick up between 11 and 14 for everyone again. And again, in terms of the adversities, we see this sort of graded association already by age 3, which persists through time, right through to age 14 at least. And then finally, for the prosocial behaviours, so you see something similar but inverted. So more prosocial behaviour is a good thing. So those who have right through to at least age 14. And when we started to look at whether these associations were being driven by particular adversities, we actually saw that most of the adversities that we looked at were associated with our 3 outcomes in terms of the rate of change over time, which suggested that all of our adversities that we looked at were important. And what I've done here is I've just pulled out the 2 that were most strongly related to each of our 3 outcomes, were most strongly related to internalizing trajectories. So it was harsh parenting on the left. So you see that this one's at age 3, but there's a bit of catch up over time with convergence over time. We actually saw where we see this difference at age 3, but this actually diverges over time. And so in the middle, we have the 2 adversities that were most strongly associated with externalizing problems. So on the left, we have harsh parenting, and on the right, we have physical punishment. And in both cases, those differences remain over time. And then finally, at the bottom, we have the prosocial behaviors, and harsh parenting and physical punishment, again, were those that were most strongly associated with differences in prosocial behaviors. And again, you can see that you see this difference at age 3, which remains through to at least age 14. So there seems to be this sort of long-term association. Okay, so finally, to conclude, early life adversities were associated with differences in internalizing, externalizing, and also the more kind of positive prosocial behaviors by age 3. Also, these differences generally persisted across time through to at least age 14 when we looked up to, but some divergence or convergence is seen. So depending on what you were looking at. So in terms of divergence, we saw this divergence for the associations between the number of adversities and internalizing problems, and also for the association between parental depression and anxiety with internalizing problems. And then we saw this convergence or sort of catch up of those who were experienced harsh parenting and internalizing problems. We also observed no gender differences in the associations, but just saw that boys were more likely to receive harsh parenting and physical punishment than girls. And then finally, what's to be gained from interdisciplinarity? So my focus as a social epidemiologist is obviously on population health. In our research group, we do some research that aims to look at how social experiences get under the skin to produce changes in health, particularly looking at things like inflammatory markers and things like cortisol at the population level. But we're really limited to the types of biomarkers that are practical and feasible to collect in large population studies. And these tend to be quite blunt indicators of what's really going on. So I think we could learn a lot more from working with people like neuroscientists like Andrew, who study and understand more about perhaps the neural mechanisms that might be involved. So the mechanisms that are far harder for us to look at using large population data sets. Secondly, also as population health scientists, we can be quite removed, I think, in many ways in terms of our work. And so we could probably learn a lot from people who are more patient or client facing. So people like psychologists and psychiatrists and social workers as well, perhaps, to understand more about the lived experiences of children who are in these types of situations as well, because I think that could really help to inform our research. And so that's everything from me, but yeah, feel free to get in touch if you've got any questions, etc. Thank you.
Thanks so much, Rebecca. That was really a fascinating start to the seminar. Now we'll hear from Eamon Mccrory next, and as I said previously, we'll have questions at the end, and we'll have all the speakers discussing them as a panel.
Thanks, Essi. So let me just pull this up. So I'm going to move back. Okay, great. Thanks, Essi, and thanks, Rebecca. So in this talk, I'm really going to focus on...
Eamon, we are seeing your speaker view at the moment, so we're seeing both slides rather than seeing the the single slide that you are talking from.
Okay.
Yeah.
So let's see if this makes any difference. Turn that off. So I don't know, is that better, Essi?
That's great. Thank you.
Apologies about that. So let's just start again. So thanks, Essi, for this invitation. So my talk is really going to focus on the neurocognitive level. And I'm going to try in a very short period, really just to provide a bit of an overview of the kinds of work we're doing in the lab, and making an argument for a social transactional approach in particular. So as Rebecca has kind of very compellingly demonstrated, childhood adversity, a range of adversities, are associated with poor outcomes in adolescence and into adulthood. And in our work, we're really trying to identify the underlying mechanisms or pathways that might help us understand the way adversity gets under the skin. And as we know, this is important given that mental health problems are increasing in children and young people across the UK. And of course, we can seek to address that mental health challenge by providing intervention to those young people. But as Tony kind of outlined, we also need to think about prevention, and childhood trauma, abuse and neglect, we know, are the largest modifiable risk factors for future mental health problems. So to tackle the challenge of mental health problems in young people, thinking about how we can use trauma as a lens through which to understand the way in which mental health problems evolve can be incredibly helpful in thinking about prevention. So our overall model in this field is really to think about how adversity shapes biological and cognitive development. So childhood maltreatment is associated with alterations and adaptations at the neurocognitive level, and these in turn are associated with changes in behavior and cognition. And we see this as essentially instantiating what we call latent vulnerability to future mental health problems. And Essi and I have developed this model of latent vulnerability, which emphasizes the way in which some aspects of brain function adapt to early adverse environments in ways that are helpful, potentially helpful for the child in that environment, but which are poorly optimized for more normative environments and increase the risk of mental health difficulties. So you could imagine, for example, a child who may have hypervigilance to threat in an environment where there's domestic violence, but how that could be incredibly problematic for the child when they go to a school environment. So people have looked at a range of brain systems implicated in mental health vulnerability, these include reward, threat and memory processing, as well as emotion regulation. And we only have time to look at one of these today, and I'm going to focus on reward processing. And this is really to act as an exemplar of the range of systems that we have been investigating. So reward processing relates to our response to positive aspects of our environment, and children who've experienced maltreatment typically show blunted response to reward in key reward processing regions, including the striatum and orbitofrontal cortex. And blunted response of the reward system has, in a number of longitudinal studies, been shown to be predictive of future depression. And there is some preliminary evidence that this also applies to children who've experienced maltreatment. And this is really important because it tells us that there is a change in the reward system that predates the emergence of depression. And we think that blunted activity of the reward system may contribute to increased apathy, reduced motivation, and that over time, that contributes to increased risk of depression. And this kind of pattern of findings in the reward processing domain are very similar to what we see when we look at threat and memory processing. We see that there are measurable changes in these neurocognitive systems in children who've experienced abuse and neglect, and that these changes are evident prior to mental health problems emerging. And that's really important when we come to prevention. And the changes that we see are in brain regions that are implicated in common mental health disorders associated with maltreatment. So by understanding and identifying these neurocognitive differences, we can be motivated to focus on prevention, because it helps us understand how and why mental health problems emerge in the first place. You know, a young person doesn't suddenly wake up one day with depression, with anxiety. Those mental health difficulties have emerged over time, and we need to understand the etiological pathway to those disorders if we're going to develop effective approaches to prevention. So in some senses, we can frame the neurocognitive research that has been carried out to date within a stress exposure model. We can think about how maltreatment or adversity leads to alterations in neurocognition that instantiates latent vulnerability, increasing the risk of mental health problems. So there's a lot of support for this, but over time, Essi and I have become increasingly aware of a major gap in this model. And that is the way in which our social world in adulthood, or indeed in adolescence, impacts our mental health. So this model doesn't really tell us very much about how, for example, neurocognitive alterations following childhood adversity can play a role in shaping the social world that an adult experiences. Nor does the model really help us think about the relational nature of mental health. It very much emphasizes the way in which mental health problems are kind of residing in the individual, that early adversity leads to these neurocognitive changes, instantiating vulnerability. And that's a problem because we know that mental health and well-being are intrinsically relational phenomena. And now when we look at studies that have investigated social functioning in individuals who've experienced childhood maltreatment, we see that these individuals are characterized by increased social stress, smaller network size, and increased loneliness. And these are features that persist well into adulthood, many decades after the experience of childhood maltreatment or abuse. And therefore, we think these are key factors in understanding how latent vulnerability emerges. And we all know, especially at the moment, how important social network is for our own well-being. And these are all factors robustly associated with mental health in the general population. So to advance our model, we need to think about not just how neurocognitive changes can instantiate vulnerability. We need to think about how they may play a role in the way in which the individual as an active agent shapes their own social ecology over time. So we can think, therefore, of neurocognitive changes associated with maltreatment as dynamic factors that are reciprocally influencing the construction of the social environment across development. And this pathway from neurocognition to an evolving social ecology is one where we have very scarce empirical evidence. So we're thinking this very much as a developmental transactional model. And the key question then, of course, becomes, well, what are the mechanisms whereby neurocognitive changes might influence an individual's social ecology over time? And we've been focusing on two different constructs, stress generation and social thinning, and I'll introduce these incredibly briefly and then say a little bit more about the work in our lab. So stress generation is a concept that has been drawn from the depression literature, and it's a phenomenon whereby individuals who are prone to depression or in remission from depression are more likely to experience interpersonal stressors than their peers. And this is a very well-established phenomenon. And the same phenomenon is evident in individuals who've experienced childhood maltreatment. These individuals are also at risk of future stressful life events. And this is important because these are events many years, often many decades after the experience of maltreatment or adversity in childhood. And in a recent study with Mathieu Guérin, who was a PhD student in our lab and now doing clinical psychology, we looked at this in a propensity score math sample where we looked at young adults some of whom self-reported maltreatment in childhood and some of whom did not, we found that those who had childhood maltreatment experienced a greater frequency of stressful life events, and that these in turn contributed to elevated symptoms of anxiety and depression. And what was really important about this study was that we propensity score matched the samples and controlled for baseline levels of symptomatology. So this is compelling evidence that something about the maltreatment experience way back in the past is influencing how the individual is shaping their social world in the present. But it's not simply a question of presenting risk, it's also about the attenuation of protective factors. As I've said, individuals who've experienced maltreatment grow up to be adults with smaller social networks and who experience greater loneliness than their peers. And we term this phenomenon as social thinning and define this as a pattern of attenuated social capital relative to peers, including the extent and quality of social relationships that emerges over time. So what the next question might be is how we then characterize the neurocognitive underpinnings of these different mechanisms. So there are a number of different candidates. We can think about threat processing or reward or memory. And at the moment, there has been no systematic investigation of how these neurocognitive mechanisms may contribute to these different processes. We can speculate, if we go back to the reward system, for example, that attenuated reward may reduce the salience of positive social cues that potentiate approach behavior. And there's one study that has looked at adolescent girls using ERP, and they have shown that blunted reward processing is associated with greater stress generation over time. So reward processing may be one neurocognitive mechanism that may be implicated in stress generation, but that has yet to be looked at in relation to childhood adversity. We also might expect that elevated apathy or reduced motivation to exert effort may compromise social interactions with peers. And we're currently collaborating with John Royster and his lab to look at apathy and effort processing in children who've experienced documented abuse and neglect. And this is a study led by Dana Ambros-Dugank and Vincent Valton, and we are using a computational paradigm where children have to exert effort by squeezing a gripper to exert effort in order to gain a level of reward. And we can manipulate the level of effort and reward in order to very sensitively measure an individual's motivation and the effort-reward balance. And what we found is that children who've experienced maltreatment in the past show higher levels of apathy, but they also show differences in neural response during effort processing. So this figure simply shows the way in which individuals who've experienced maltreatment show reduced activation of the anterior cingulate cortex during effort exertion relative to peers. And we're following these children up over time, and we're really interested in whether these differences in apathy or in effort processing are related to future symptoms, as well as whether they're related to differences in how those children are engaging and interacting with their peers. We're also looking at other potential mechanisms and processes that may relate to social thinning. So we're looking, for example, at facial trust processing, and this is a study led by Lenny Neal, where we look at a scope of faces that vary in trustworthiness based on a prior study. And we ask children in a forced trust task to identify whether this person is trustworthy or not. And using, again, a computational model, we're able to build a picture as to whether individuals who've experienced maltreatment are attributing trustworthiness to unfamiliar faces. And what we found is that individuals who've experienced maltreatment typically show a reduced attribution of trustworthiness to unfamiliar faces. And we can imagine how that might then impact their ability and propensity to cultivate relationships with new children and with new peers. And again, we're following these children up over time to see whether these differences in trust attribution are influencing the way in which these children function within their peer networks in the future. So where next, if we think about where we need to go in terms of interdisciplinary collaboration, I think it's clear for us at least that it's not sufficient to simply document brain changes following stress exposure and relate these to symptom domains. We also need transactional mechanisms that shed light on social determinants of latent vulnerability. So we need collaboration between social scientists as well as computational modelers. If we're really to understand the way in which these neurocognitive changes play out in terms of the social context. And such collaboration, I think, is essential if we are to make progress on prevention. So I just want to thank everyone in the lab, all of our postdocs and research assistants, young people and families who've contributed, and in particular Essi Viding, who I will now pass back to, who's a collaborator on all of this work.
Great, many thanks, Eamon, for this extremely clear talk. And again, reminder, we're saving questions to the end, and I see a number of quite specific questions. We will ask some of those if we have time, but I would really encourage people to think about more cross-cutting questions where all of the speakers could contribute to, and perhaps email the speakers about very specific study detail related questions. So our next speaker is Andrew Macaskill, who is doing animal neuroscience research and who will be talking to us about early life adversity and value-based decision making in mice.
Okay, can everyone hear me, hopefully? Thank you very much for that introduction and for the amazing 2 talks that came before. So as you said, I'm based in the Department of Neuroscience, Physiology Pharmacology. And so I'm a basic neuroscientist studying these kind of problems in rodents. And so hopefully what I'm going to try and take you through today is hopefully some little example experiments and some of the kind of thought process behind what we're doing, so to give you an insight into kind of hopefully what we can offer and what hopefully we're hoping you guys can offer us too. And so as we've heard from the previous 2 talks very eloquently, it's becoming clearly obvious that adversity during early life, such as stress, and particularly during the critical period of adolescence, shows a marked increase in the risk of developing mental health issues later in life and in adulthood. And as a basic neuroscientist, what I'm fascinated by is the fact that we still do not really understand what this early life adversity actually does physically to the brain that results in this increased risk. And an increasingly prevalent hypothesis is this idea that Eamon kind of introduced, which is that early life adversity actually seems to preferentially alter neurons and circuits that are that underlie the ability to carry out what's called value-based decision making. In essence, the ability to understand and choose an action based on how good or bad you think it's going to be. And consistent with this idea, the ability to carry out value-based decisions, again, as Eamon said, is actually very much associated with the risk of developing mental illness, to such an extent that in a number of cases, the severity of a deficit in value-based decision making is actually correlated with the severity of the disease. And so, as I said, I'm a basic neuroscientist, though. And so what I'm very interested in is using model systems, and in particular the mouse model system, to really understand how early life adversity can alter neurons within the brain to result in these differences in value-based decision making. And in particular, we're interested in 2 sides, one of which is to really draw the types of tasks and behaviours that we're studying directly as directly as possible from human studies investigating mental health. And also to try and bring in the kind of early life adversity that we're studying, again, from the beautiful stuff that Rebecca was talking about earlier, from epidemiological studies identifying specific risk factors. And so what I'm going to talk to you about today is just a quick example few experiments where we're utilizing early life and adolescent social isolation. So this is where we isolate mice from their social peers from puberty. And we're using that as an early life stressor and looking at how this changes neurons within the brain to result in deficits in value-based decisions based on reversal learning. And so what I want to quickly just go through is to go through what reversal learning actually is, so we're kind of all on the same page. And so as a mouse scientist, we're always using mice. And so for a mouse, a reversal learning task is simply if it's in a behavioral chamber and it is presented with 2 levers. Okay, and from the start of the trial, the mouse can just choose to press either of these levers. And if the mouse presses the left-hand lever, it will receive nothing. It will just get a timeout and will be pretty frustrated. If it presses the right-hand lever, it will get a nice sugary liquid reward. And the key to this task, though, is that sneakily, without telling the mouse, I will then change the contingency of this task such that suddenly the left lever will be the lever that will give you the nice sugary reward, and the right lever will now not give reward. And these contingency changes can happen multiple times across an hour-long session. And so within this kind of reversal learning paradigm, the goal of the mouse is to work out from a trial and error kind of strategy which is the best lever to press at any one time. And this is kind of a classic value-based decision-making task. And as I said, the adversity we're using as a model system here is social isolation during adolescence. And so what this means is we're going to take a litter of mice, and for half of them, we'll rear them in a social group. And for the other half, we will isolate them from puberty. And this is a potent stressor, both for rodents, but also for humans. And so what I just want to first show you is what a control mouse looks like, our group house mouse looks like when it's doing these tasks. And so what I'm showing up here, this is just when this gray bar is at the top, this means the left lever is getting the reward. And when it's at the bottom, it means the right lever is getting the reward. And this gray trace here is just the choices of the mouse over time. And all I want you to do is take away just two things from this. The first is that when the left lever is high, the mouse tends to press the left lever more times. And when the right lever is high, the mouse tends to press the right lever more times. Okay, so it really can carry out this reversal learning pretty successfully. The second thing I want to take away from this is the fact that it is, the mice are remarkably good at this. In fact, if you look at these switches, the mouse is only really taking 1 or 2 trials in order to be able to understand that the contingency has switched. Okay, so that's what it looks like for control mice. And so this is actually quite similar to the way humans would carry out this task. What happens when we socially isolate the mice and then test these mice in adulthood? And what you can see, hopefully, from this graph is that just generically, the mice are just not as good at carrying out this task. And this difference really seems to be in the fact that they are very, very slow to realize that a switch has happened and to update their action appropriately. And there are various ways to quantify this, but we've been working a lot with some computational psychiatrists to develop very simple computational models that allow us to understand the actual deficits that are occurring in these tasks. And we're doing this in a number of ways, but just for simplicity, I'm showing the simplest version of the model here, which is a Q-learning model that just shows that these differences in behavior that I showed you can be attributed to a reduction in the learning rate of mice during this task. This is quite exciting for us because, in essence, what it shows is that it recapitulates a lot of studies in humans showing that social withdrawal and social isolation in early life actually results in very similar deficits. Okay, so up until now, all I've shown you is that we can do exactly what has been already found out in humans in mice. And so what I want to spend the next few minutes on is just telling you actually why we're interested in doing this. And obviously the reason why we're interested in doing this is to actually understand the cellular and circuit mechanisms underlying these changes in behavior. And so just for an example, I'm going to focus on a part of the brain that we've had a lot of interest in my lab recently, which is the hippocampus. And so the hippocampus, which I'm showing here in red in humans and also here in red in rodents, and it's very homologous across the two species, is very much implicated in the neural alterations associated with early life adversity. And so we've spent the last few years really analyzing and investigating this circuit in detail. And as a result, I've actually identified the hippocampus is made up of a large number of very distinct populations of neurons. And so just to exemplify this is just a 3D image of a mouse brain where we've color-coded the neurons in the hippocampus according to 3 criteria. Just one population projects to the prefrontal cortex, a second population projects to nucleus accumbens, and a third population projects to lateral hypothalamus. And what I want to take away from this is the fact that these are all individual populations of neurons, and we're increasingly finding that these carry out very unique roles and perform specific calculations during behavior. And so what our real goal is now to use is to use this kind of ability to really understand the makeup of the hippocampus, to understand how how the brain is actually being altered by social isolation and early life adversity in general. And so I just want to give a quick example of the kind of things that we can find with this. And so what I'm showing you here is now using this ability to label specific populations of neurons, we're now making electrical recordings from a specific population of neurons, in this case, the population that projects the prefrontal cortex. We can then stimulate these neurons and look at how excitable they are by measuring the number of action potentials that they fire. And so what I'm showing you here is just a graph for control mice, showing that as we increase the amount of stimulation we give these neurons, as you'd expect, they increase the number of action potentials they fire. But what is very interesting is that if you do this exactly same experiment in mice that have been isolated in adolescence, you see a dramatic decrease in their excitability, suggesting that social isolation causes this decrease in activity. But what makes us extremely interested by this result in particular is that if you record from any of the other populations of neurons that I showed you in the hippocampus, they're not affected at all. And what this really suggests is that social isolation is causing a very specific change in a very specific type of neuron in the hippocampus. And we got quite excited because we thought that potentially this may give us a lot of information about what's actually happening in the brain. And so just quickly to summarize all that data, I've shown you that social isolation in mice results in specific deficits in the learning rate during probabilistic reversal learning. But I've also shown you that hippocampal neurons that project the prefrontal cortex, this very specific population of neurons, are specifically inhibited by adolescent social isolation. And so this is tempting to think that this deficit in neural activity might be causative for the changes in behavior. But at the minute, all we've really done is look at a kind of correlation. So we know that social isolation causes both of these things. What we really want to ask now is, does a reduction in the specific population of hippocampal neurons actually result in a deficit in behavior? And by taking advantage of a technique called optogenetics, we can actually in mice do exactly this. So we can actually, and so in a nutshell, what optogenetics is the ability to remote control the activity of neurons in real time, so with millisecond precision, simply by shining light on the neurons. And so what I'm just showing you here is a schematic of a neuron recorded by a PhD student in my lab, Karina, where she has infected or expressed this optogenetic construct in this neuron, and she is stimulating it, and you can see that stimulation causes a nice action potential firing, as I showed you before. But now she's doing exactly the same thing, but now shining a brief pulse of red light during the stimulation. And you can see that the red light now beautifully blocks all the activity of these neurons. And what this technique allows us to do is the causative experiment. And so we can now take control mice that have been reared completely normally all the way through into adulthood, and then use this construct to specifically inhibit only this one population of hippocampal neurons only during the reversal learning task. So that the only thing that's changed between the control group and the inhibition group is the activity just of these neurons. We can then allow the mouse to carry out the reversal learning task and look to see what the effect of this inhibition is. And so when we do exactly that, what we find is quite exciting. And if you just focus on this graph here, it's that we actually can almost completely phenocopy the behavioral deficit of social isolation just by inhibiting this one population of neurons. And just to show you what it looked like with the actual social isolation, you can see that it is essentially very, very similar. Okay, so just to start to wrap up, I just wanted to quickly go through what we're currently doing and what we're thinking. And so one of the key drives in the lab at the minute is to utilize computational psychiatry. And so we're working a lot with people like John Rozier, Quentin Hughes, and Annie Talwar. And the idea here is to try and use computational psychiatry to build a bridge across species and really to allow us to implement the kind of knowledge we're getting about the circuit and systems neuroscience in rodents to more human and clinical mental health research. And that would be a kind of maybe a ripe thing for discussion later. And we're also really interested in comparing multiple dimensions of early life adversity, both individually and in combination. And this kind of gets back to the thing that Rebecca was talking about at the start with ACE scores that simply add up different forms of adversity, but also taking into account specific combinations and modes of adversities and looking at their commonalities and distinctions. And then I guess I just wanted to quickly think, well, why are we actually all doing this? Take a step back and stop getting all excited about neurons, because that's kind of tend to what I do as a neuroscientist. And the real reason I'm interested in this is that it's become increasingly clear that diverse different adverse events, or even the same adverse events in different individuals, may result in a unique combination of circuit alterations. And in the future, the long-term goal for us would be really to be able to try and create behavioral markers that point to specific circuit deficits. And this would allow us, hopefully, to move beyond drugs targeting entire neurotransmitter systems, which we're kind of utilizing now, and towards more specific circuit-based treatments, and towards kind of more personalized treatment based on a circuit understanding. And so this is why I'm so super excited about this kind of the Mental Health Institute and the Child and Young Person Mental Health series. And because we are lacking so many things and really want to integrate as much as we possibly can with the wider community. And so just as a kind of some random thoughts, like from a population and intervention perspective, we really want to know people's thoughts on whether we're investigating the right modes of adversity, whether we're doing these in the right combinations and at the correct times during development. And we're really wanting to draw from that literature as much as we possibly can. And from a human neuroscience perspective, we're also very much interested in specific experiments or experimental designs that we could do and adapt to allow us to more directly compare to human data sets. And then finally, I also wanted to just put out there is like, maybe as a discussion topic, is that do we really think that behaviors that are technically simple enough to be able to be carried out in rodent systems, such as mice or rats, are actually enough to be able to kind of get to this stage where we can translate across between systems and clinical neuroscience. And so with that, I just wanted to congratulate all the people who managed to achieve all this crazy work, and in particular Candela, who did all the physiology, Karina, who did all the behavior, and Ryan, who did all the anatomy.
Thank you very much, Andrew, for a really fascinating talk. I really want to begin the panel discussion by thanking all the speakers. I'm sure I'm not the only person who it has been really thought-provoking to think about this topic from very different perspectives. And I also want to thank the speakers for really trying their best to think about how their research might benefit from connecting across disciplines. So I think that there are already some really, really interesting ideas there. I can see that in the chat, the speakers have very kindly replied to some of the more specific questions in the areas, but there have been also some cross-cutting themes. So I'm going to sort of start off the discussion between the speakers by really sort of wanting to hear all of their thoughts on how each of the speakers think you might best translate from this sort of group level or individual differences level data to actually single individuals. So what I hear from speakers and also is coming across in the questions is that there is huge variability. So in a group level, there is clear impact of these adversities, but the impacts are not same for everybody, and the trajectories following the impacts are quite different, and they may depend on individual differences in genetic vulnerabilities, individual differences in other environmental factors that are present or not present. And you can have this phenomenon called multifinality, where you have certain commonalities, but how they actually canalize our development may be quite different. And of course, in a therapeutic context, we want to think about individual prediction, not just prediction at the population level. So it's quite a conundrum, and it would be really good to hear from all the speakers how they think we might go towards resolving this, and whether this will necessitate connecting across disciplines. And Rebecca, maybe if you can start. I think Rebecca looks like she's possibly frozen. So let's get Eamon to start, and we hope to have. Oh, you are back. You're back.
Oh, I'm back. Sorry. Yeah, ongoing problems today. I think this is a really important point about, and it comes up so frequently, about the differences between looking at these things at a population level. And so I was very careful at the beginning about how I described the sort of prior evidence to say, like, this is just like population average. We cannot just necessarily disaggregate this to individuals. And I think we wrote a review paper last year that really brought out this issue, which I sort of call Epidemiology 101, which is like, we're talking about this higher level kind of averages rather than individuals. And most people who experiences these types of adversities we've looked at actually don't go on to develop mental health problems. And Jessie Baldwin wrote I think she's here actually, wrote a really nice paper that's just come out in the last week or so, which shows this sort of the differences between individual prediction and this sort of group-based, when you're looking at things like ACE scores, that most people that have an ACE score kind of don't go on to, and I think that's just important to remember. I think there's so much of that kind of gets lost in this sort of ACEs discourse that's really become very, very popular recently. And yeah, I think it's important to remember that that we're talking about, and certainly in the work that we do, we're talking about the sort of population level. Yeah, so.
Eamon, do you have any thoughts?
So I completely support everything Rebecca said and really highlight that paper Jessie has just published. I think you're asking the $1 million question, Essi. So thanks very much for giving us an easy start. In some ways, there's so many different domains that play out, I think, in relation to the individual. So someone in the chat referred to subjective experience. So we know that's very powerful. The same experience, as Andrew's just said, can be experienced very differently by different people. People have different underlying genetic vulnerabilities that will interact with that experience. There can be timing issues about when the adversity occurs in relation to brain maturation and social maturation. There's also the context in which it occurs, and then the protective factors that are there or aren't there. So it's a highly multifactorial, complicated equation. You know, what Rebecca and I are doing is like we're aggregating at the group level, Rebecca at a much bigger group level, but I'm still aggregating at a group level. So you're asking us for answers, Essi, not the problem. We're just restating the problem, so apologies for that. But maybe what I would pitch for, and I think this is something you and I have discussed, is that we need, from a cognitive neuroscience perspective, much more sensitive measures able to capture individual differences that are normed, that are reliable, that are valid, that are able to track something, say, like threat reactivity at, say, a 2-year-old, a 4-year-old, a 10-year-old, that there is reliability and internal consistency across development in that measure. So we need, and computational psychiatry may be one approach that can help improve our capacity to refine measurement of the key neurocognitive systems we believe to be implicated in disorder. There are other things I could say, but I'm going to pass over to Andrew now.
Yeah, I think it's, well, so from my perspective, it's, yeah, the perfect question. Because again, like Eamon and everyone, as basic scientists are super guilty of just putting group A versus group B. But I think what I would say is that in particular, a number of labs in basic science are starting to realize this as well. And we actually do have quite an opportunity to try and maybe address this using basic science. And so, for example, when I was hinting at this idea of multiple forms of adversity in combination, the idea is we're actually going to try and set up reproducible and standardized conditions so we can create large data sets of which we can make multiple different manipulations and start to look at mouse to mouse or rat to rat differences, irrespective of group. So we will have experimental groups, but then we can use, for example, hierarchical clustering analysis or some really nice things to look at different forms of resilience. And the nice thing about basic neuroscience is we can do multiple behavioral tests in the same mice and then also look at that mouse's specific neuronal alterations. And so we're maybe moving towards a kind of more behavior and biology based approach rather than a categories based approach, and then trying to relate these things back to each other might be a kind of a way we can really try and do that kind of in a quite controlled way from the basic neuroscience perspective.
That's really interesting. And I think almost a follow-on question from this is if we think about the point of this seminar series and also the mental health strategy and what the Institute of Mental Health is trying to accomplish more broadly, I think it would be really helpful to think, how can we foster dialogue between the disciplines so that, you know, if you as an animal researcher could think about, well, how does human cognitive neuroscience or human population health, how does it help you to limit the problem space that you have when you think about these individual differences models? And similarly, I guess, for the human neuroscience or population health scientists to think about, well, what would they want from animal studies in order to help them limit the problem space in their research? And I think that that's a kind of a challenge for us as we go forward. I'm going to ask one more general question, which is, what do the panel members think about how critical it is for us to develop very clear common language and agreed concepts in order to do this back and forth translation. So I know from the field of behavior change, for instance, there is some very interesting qualitative research which suggests that often scientists, they think they're talking about the same thing, but they actually are not. And I'm wondering to what extent you think this is preventing effective crosstalk and collaboration across different disciplines. But Andrew, do you want to weigh in first?
I have a terrible admission to make that I probably shouldn't really make. I'll make it anyway. Oh well.
Please.
My reputation's gone anyway, isn't it? Anyway, but yeah, so I don't think I really know what internalization and externalization actually means. Like I can kind of guess. So that's quite a good example, I guess, right? And I'm pretty sure I waffle on about insane like biophysics and stuff a lot of the time and have no idea what I'm talking about. But like, yeah, I think that's a definite issue, especially when you, I think it's really cool listening to what Rebecca was saying earlier, because it's so pertinent to what I'm doing. But I find the literature like super hard to get into simply because they use a lot of long words that I don't understand. And it's kind of crazy that we're in the same field but aren't are kind of at cross purposes sometimes. That's a really, really good point, I think.
Can
I respond to that? Yeah, I feel exactly the same, but from the other perspective. Like, why are we not talking to each other? But also, I don't understand like the papers that you write, but we do the same type, well, not the same type of research, but we're looking at the same kind of questions, but from different perspectives. So I think it is a problem for interdisciplinarity. And I think in social epidemiology, in some ways, we're inherently interdisciplinary, but we sometimes have the criticism of being kind of, we take bits from lots of different disciplines, but we're not really a master of any of those things. So I'm not a psychologist, so I don't know the ins and outs of like what people might present with, for example, in a clinic. But yeah, so I think it's useful to take a step back and think about, we could even call the statistical methods like under different names, but actually we're doing the same thing. Yeah, so I think this is, I feel it as well, basically is what I'm saying.
Yeah, so we actually, for example, have a mouse ACE equivalent that we're trying
to,
so it's like we can actually
Who knew?
but it's called just like, something stupid. So yeah.
So we probably need a workshop where we sort of do a interdisciplinary concept paper on the concepts or something like that.
Yeah, we need a glossary or something.
We've got a few questions that are a bit more specific, but I think lend themselves to at least a couple of speakers here in the chat. One of them is about our education system and the concern that this causes a lot of stress to the children because there are a huge amount of assessments. And the person asks whether this constant stress for children with ACEs is something that keeps their cortisol at harmful levels and causes them to find daily life more difficult than their neurotypical peers or peers who have no ACEs, and whether this is reflected in the research or what the thoughts are, particularly Rebecca and Eamon, what your thoughts are regarding this issue.
Yeah, I think this is an important thing, and I think this has been a concern for a while, and I feel it as a parent as well. It's but I don't know that we know, I don't think anybody's looked at it, and I think it would be good to look at it. Whether we have the data to do that is another thing. So we often, so we've got some good like adversity data, but whether do we have the information about how stressed the child feels about specific situations at school? I'm not sure that we have that as well in the same data set. So it's a great idea. I'm not sure that we know the answer to it, but on the flip side of that, we've been doing some work that's looking at kind of more positive things at school and whether kind of more positive things at school can be a potential buffer for early life adversity. So in some ways, we're kind of looking at a bit of a deficit model at the moment. We're just looking at mostly the negative things, but actually it'd be good to also look at the positive stuff as well around schools. But I totally take the point about testing but whether we can look at that is another thing, because we need that data in the same kind of data set. But yeah.
Eamon, do you have anything to add?
I just agree with everything Rebecca said, but maybe just to add that I think one of the real challenges in trying to address that question is the fact that all the kids are in school and all the kids have exams. So it's not a factor you can kind of manipulate or find half of the kids in the country who aren't doing exams. Because at different ages that might be possible, but with GCSEs and A levels that are the most salient exams, they're going to be there, so it's going to be hard to disentangle the impact. But I think, as Becca said, there's just an importance also of thinking about how to buffer. So we know that childhood adversity will make kids more susceptible to stress, and that goes throughout the lifespan. So we need to think about, well, how do we develop the coping capacities and the skills that those young people need? How do we foster their ability to make peer relationships that can help buffer those stressors? Because stressors will happen when they become adults and will be ongoing. So it's really about how we can build those factors that can promote resilience, as much as thinking about removing stressors that are probably to some degree inevitable in any educational system. But it's not to say that they couldn't be modified.
And I think cross-cultural research might be interesting in this context, where, you know, from different countries have different systems in terms of, you know, how they examine children. I'm very conscious of time. There's one more question I would like to be considered, and then I'm going to make some closing remarks. So there are a couple of people who have queried about the value of disciplines such as anthropology in doing this research and trying to understand about subjective experiences and cultural variations. And I'm assuming it's a quick thumbs up from the human researchers. I guess mice don't have that much cultural variation, but maybe they do. Secret life of mice.
Yeah, there is a person who's convinced that Ukrainian mice are much less susceptible to anhedonia than not, but I'm not sure how real that is. So
I love that. That's great. That's I think we have time for one more question, which is again reasonably specific to the human presenters. But there's a question about how much increasing public mental health awareness might influence sort of people's self-recognition and parent reporting of the symptoms. And do we, you know, maybe this is most for Rebecca in terms of, you know, if you look at different cohorts, do you expect to find different patterns because you have differences in the awareness, for instance?
Yes, definitely. I mean, for example, just giving an example, so we in the Millennium cohort study that I've shown here, we've got items that are specifically about smacking as a form of physical punishment, but we wouldn't have had those in earlier cohorts because it just wasn't something that you necessarily thought was important. So I think, you know, the changing cultural context is so important, and as a life course researcher, that's something we think about a lot. Yeah, so things change over time. The reporting of things changes over time. I suspect if you ask these kind of questions now about smacking, that people would feel quite embarrassed about reporting it, would likely really underreport that now. Yeah, it changes. So it's very important.
Okay, I'm going to sort of close now because we're very nearly to time. But I wanted to sort of first say that I really hope that these seminars will become a real vehicle for consolidating and initiating collaboration in children and young people's mental health research across UCL, and also help us to drive forward innovation and fresh thinking in children and young people's mental health more broadly. I want to highlight that we've got a new website. It's only just been launched, and there will probably be some teething issues and we'll be fiddling with it, but it's up and running. It lists our research, our education and training and other activities. It has some videos up and running, and we're hoping to update this and make it more user-friendly. And we'll also hope to collect feedback from the community and make changes based on that feedback, so that the website can best serve us and also serve the outside world that we're trying to reach. Next seminar in the series is 10th of March at the same time, and this is on loneliness and children and young people's mental health. Again, another interdisciplinary event, and we'll post information about this and further seminars shortly. Finally, I really want to say a huge thank you to Children and Young People's Mental Health Strategy Implementation group members who've worked really hard in setting up this seminar series. And these include Pascal Fearon, Jess Dayton, John Roycer, Pravitha Patalay, David Osborne, Sonia Johnson, Jane Hurry, Ross Shafran, David Scoos and Quentin Hughes. So we've had a really amazing team of people brainstorming ideas, and this seminar series wouldn't have come about without them. I also want to thank again the Neurosciences domain who has sponsored this, and both Neurosciences domain and Institute of Mental Health who've supported development of the seminar series. And finally, thank Matt Wakelin, Maya Sophia, Maria Thomas, Antonia Maiola and Anna Phillips, who've all been brilliant in setting up this event and the communications relating to it. And I hope that we will have most of you back for our subsequent events, and please feel free to email myself or Matt Wakeling with suggestions for topics and speakers. And we hope we're going to really be able to showcase a wide range of research over the coming months. So thank you everybody very much for coming.